here's your babies in 4D. And as we can see, they're identical.
Twins are as close as nature gets to human cloning. Their identical genetic makeup is one of the wonders of human reproduction. The close similarities can astound. Even identicals separated at birth often lead strikingly similar lives, with the same jobs, tastes, and hobbies. But identicals can also confound expectation. Advances in genetic science are revealing that identical twins are not as identical as we thought. Through state-of-the-art in-the-womb 4D ultrasound scans, scientifically accurate visual effects, and specially shot microscopy footage, we will reveal the hidden world of twin fetal development. In a world where most of us are individual, identical twins capture the imagination. But for scientists, it's not their similarities that prove most fascinating, but their differences. They may not look it, but these boys are identical twins, born with dramatically different birth weights. Twins like these, with major physiological differences, present an enormous opportunity to reveal truths about human identity. From fingerprints and intelligence to disease and even sexual orientation. Like everyone, identical twins begin life as a single cell. A fertilized egg called a zygote. This cell contains 23 pairs of chromosomes, tightly packed strings of genes, one set from the mother's egg and one from the father's sperm. Together, they make up the entire genetic blueprint for a new human life. A few hours after fertilization, the cell, with its cargo of chromosomes, embarks on a remarkable journey. It travels along the fallopian tube towards the womb. The single cell now starts to divide. First 2, then 4, then 8. Each cell an identical replica of the other. This rapidly multiplying collection of cells is called a blastocyst. And it's here that nature's most remarkable reproductive anomaly can sometimes occur. Very occasionally, there is an event so mysterious that until recently, it's never been witnessed. Several days after conception, the blastocyst spontaneously splits into 2. Each new blastocyst is composed of cells with the same identical set of chromosomes, carrying the same arrangement of genes along their length. The 2 blastocysts now have the potential to develop into identical twins. Exactly how and why the blastocyst splits is one of reproductive science's greatest mysteries. But in 2007, during a laboratory study of 26 embryos, embryologists witnessed this event for the first time. The blastocyst's outer membrane is called the zona pellucida. The lining inside is called the trophoblast. Just one cell layer thick, the trophoblast will eventually develop into the placenta. Within lies the inner cell mass, embryonic stem cells that will eventually become the fetus. Over the course of 4 days, the embryologists noticed that a junction between a cell and the trophoblast membrane would regularly weaken, letting liquid leak out and causing the trophoblast to collapse, like a balloon full of water. Then it would repair itself. But in 2 of the eggs, the embryologists spotted something no one had ever seen before. After the membrane collapsed, the inner cell mass divided in 2, each clump sticking to a different side of the trophoblast as it reinflated. If these clumps were to develop in utero, they would eventually grow into identical twins. Born from the same single fertilized egg, it's not surprising that most identicals retain a similar physical appearance as they grow up. The influence of genes reaches beyond how we look. Genes also affect personality and taste, making us flamboyant rather than shy, with a taste for coffee instead of tea, and a love of routine or desire for experimentation. Similarities between twins are often more than skin deep. Dennis and David Herrera are 53-year-old identicals from California. After high school, both became officers for the Los Angeles Police Department. Because they were raised together and went to the same school and had the same friends, it's impossible to say whether their career choice was influenced by their genes or their upbringing. To really test the extent to which our genes influence our life choices, scientists study identicals who have been separated at birth and raised in different environments. Daphne and Barbara were separated as babies and had no contact with each other for 40 years. They were raised in very different families, one by a scientist, the other by a municipal gardener. But the similarities between them are striking and perhaps beyond coincidence. They both left school at 14. At 16, they both fell in love. In their early 20s, they each married their childhood sweethearts. They both suffered miscarriages at the same age, but went on to have children in their early 20s. Both have an irrational fear of blood and of falling down. When they met for the first time on a train platform, they wore almost identical clothing. The similarities are remarkable. They suggest that genes play a powerful role in shaping many aspects of our lives and personalities. But as we will see, it's the differences between identicals that could offer science the greatest opportunity, the chance to solve the age-old debate of nature versus nurture. When these twins were born in 2006, Jake weighed a healthy 2.5 kilos. His twin, Tom, weighed just over half a kilo. Since they're identicals, this dramatic difference was a mystery. For answers, doctors looked to the one environment the boys shared before birth: the womb. By day 5, the two identical blastocysts have reached the womb. Now they face one of the most critical events in their 9-month odyssey, one that could dramatically affect their fortunes, in the womb and beyond. When they were born, Jake weighed 2.5 kilos, and his brother Tom, 0.5 kilo. And yet they're identical, born just minutes apart. How could twins with the same genes look so different? The answer may lie not in their DNA, but in events that occurred in the environment they shared, the womb. And the first, most critical event is implantation. Around day 6, the blastocyst must implant in the wall of the womb, securing its position for the next 250 days' growth. But timing is everything. Each twin will rely on a placenta to supply it with oxygen and nutrients, but the placenta only starts to develop after implantation. So blastocysts that split early, before they implant, each get their own placenta, but those that split late, after implantation, must share one. A shared placenta may mean one twin receives less nourishment than the other. As a result, it grows much more slowly, a condition called selective intrauterine growth restriction. There are even rare instances when identical twins have their own placentas and yet develop this condition. Identicals Jake and Tom are one such case. Their different weights are the result of events that occurred long before they're born, but their effects may last a lifetime. Once implantation has occurred, embryonic development is surprisingly rapid. By the end of the 3rd week, a rudimentary body shape is already emerging. The embryo already resembles the beginnings of a body, totally unprotected by skin and bone. On one edge, stem cells flow over and are assigned their fate by the organizer. One of the first organs to form is the heart, until now, just a clump of muscle cells. Then around day 22, a single cell in this dormant clump of muscle cells spontaneously contracts. It sets off a chain reaction. One after the other, neighboring cells begin to contract until the entire vessel is pulsating. Two tiny hearts, about the size of 2 pinheads, beating together. Our twin embryos have approximately the same number of cells, the same DNA, and yet, incredibly, they may already be different. Is personality innate or learnt over time? Why do some of us succumb to disease while others remain immune? And from where do we get our skin color? What makes one person gay, another straight? The answers are not as simple as we once thought. Remy and Kian are fraternal twins. The result of two separate sperm fertilizing two separate eggs. The girls' parents are of mixed ancestry. This usually means the children will also look mixed race, but not always. Being mixed ancestry means carrying a mixed set of genes for both lighter and darker skin. In Remy's case, a sperm with a set of genes that produce darker skinned children fertilized an egg with similar characteristics. In Kian's case, a sperm with genes that create lighter skinned children fertilized a similar egg too. The result? Twins. But twins who look very different. Remy and Kian show us that twins can often confound expectation, but they're not the only ones. In 2001, a pair of twins were born who at first appeared to be a boy and a girl. On closer inspection, it became clear that one had signs of both male and female genitalia. The twin was a hermaphrodite. Scientists believe that this happened because a single egg was fertilized by two sperm, one with a female sex chromosome and one with a male sex chromosome. The egg developed into a blastocyst with both male and female cells. The egg then divided, and each of the two new cells shed an extra chromosome, resulting in a blastocyst composed of both male and female cells. That blastocyst then split to create twins. By chance, one had more cells with the male sex chromosome and developed into a boy, while the other had similar numbers of cells with male and female sex chromosomes, resulting in a child with male and female genitalia. It was a startling discovery. Twins who are halfway between identical and fraternal, the only known case of semi-identicals in the world. The simple classifications we're used to are breaking down and will come to inform much of our newer understanding about how twins, and by extension all of us, develop. At 5 weeks, our identical twin embryos are beginning to take shape. Now curved into a C shape, their heads and tails can be distinguished, as can hearts, spinal columns, and the beginnings of tiny limb buds. At this stage, the embryo looks almost indistinguishable to any vertebrate in the animal kingdom. These fragile creatures, just 1.5 millimeters long, are about to embark on a critical period of brain development. This 5-week milestone could be another stage where identicals develop key differences before they're even born. Celso and Jesus are identical twins. They were raised together. As they grew up, they remained physically similar, but their tastes and interests began to diverge. Celso became interested in dance and academia, while Jesus preferred sports. The most surprising difference between the two brothers is that Celso, here wearing black, is gay. The differing sexual orientation of identical twins is an opportunity to investigate one of science's most controversial questions. Are people born gay? As Celso and Jesus were raised by the same parents in the same household, they inevitably shared the same environment at a crucial time in their personal development. In the general population, the chance of someone being gay is less than 5%, unless you have a gay twin. Here, the chances are much higher. If you're fraternal, sharing half your genes, there's nearly a 25% chance that you will also be gay. If you're identical, sharing all your genes, there's roughly a 50% chance that you will also be gay. This suggests that there must be some genetic component to our sexuality. However, it can't be all down to genes, otherwise all identicals would be either both gay or both straight. Some other factor must be at play. In their first few weeks, all fetuses develop along similar lines. If nothing changed, we would all be born female. Fetuses with the male Y chromosome will form testes at about week 6 that begin to produce the hormone testosterone. But at about the 8th week, testosterone is released and may affect early brain development. This hormone masculinizes the body. Testosterone also masculinizes the brain, including a part called the hypothalamus, part of the network which controls who we find sexually attractive. Some scientists believe that the more the hypothalamus is exposed to testosterone, the more it sets the stage for a sexual inclination towards women. Occasionally, a male fetus may not produce sufficient testosterone, or its brain does not absorb enough to shape it along heterosexual lines. If this theory is right, then it may be that Celso, the gay brother, absorbed enough testosterone to masculinize his body, but not enough to fully differentiate his brain. As a result, he was left with a desire for men. Although there are still many mysteries, twins like these are playing a crucial role in informing scientists about how and when we all develop our sexuality. By the 8th week, our identical twins have finished their embryonic stage of development and are now known as fetuses, from the Latin meaning offspring. The twins, now secure in their own sacs but sharing a placenta, have approximately 200 days left until birth. Now their growth accelerates. By 13 weeks, they're beginning to look more human. The eyes have moved together, and the head is in better proportion to the rest of the body. It's also the end of the first trimester, the time most pregnant women will have their initial ultrasound scan. The ultrasound sonographer checks for basic signs of normal development.
So here's your babies in 4D, and as we can see, they're identical.
The sonographer sees a single placenta, a sure sign that the twins are identical.
We can see here a single placenta, like a T-section, which proves that you're having identical twins.
So when would you see them start interacting or reacting against each other?
We can see them almost sort of moving.
From now on, the mother will be monitored closely. Twin pregnancies are more at risk of premature births. Immature development of lungs and organs can lead to complications. These complications make natural childbirth more dangerous. As a result, twins are around 50% more likely to be born by caesarean section than singletons. The timing of the scan also marks the start of the second trimester. The second trimester, from 12 to 24 weeks, will see not only dramatic growth, but massive development in everything from the structure of the brain to the refinement of facial features. By the end of this stage, the fetuses will be small, but almost perfectly formed. They look very similar, but new research reveals that even at this early stage, identical twins may already be less identical. Subtle changes are underway that will become greater throughout their lives. And the mechanism responsible is one of the most complex yet fascinating processes in the whole of human development. "15 weeks into their journey from conception to birth, our identical twins begin to move for the first time. They start to interact, exploring their environment with their hands and feet, touching and even appearing to kiss. Their mother in later pregnancy will feel the same kicks each twin feels in utero. At times they appear aggressive, at others almost caring. They look identical, but unseen influences may already be taking effect, ones that could subtly alter the expression of genes in their growing body. Identicals Jake and Tom are now aged 17 months. Jake is 8 centimeters taller than Tom and 4 kilos heavier. When they were born in 2006, doctors attributed these discrepancies in size and weight to restricted nutrition or blood flow in the womb. They expected Tom to catch up. But after a year and a half, they're investigating whether Tom may be suffering from a genetic condition called Russell-Silver syndrome, a growth disorder. One of the causes of this syndrome may be a problem with a growth gene on chromosome 11. Scientists are puzzling over why Tom and his brother do not both have the syndrome. Identical twins, after all, have identical genes. Genetic puzzles like these are opening up a little-known world at the outer reaches of science, epigenetics. This growing field of biology may help explain the differences between twins like Tom and Jake. Epigenetics reveals that while their DNA code may be the same, the way it functions can differ. The human genome contains around 25,000 genes, each with its own specific function, such as producing energy or directing cell division. Now, geneticists are investigating a previously unknown aspect of the genome called the epigenome. There is a series of chemicals that act as switches, capable of activating or deactivating individual genes. One of these switches works by a process called DNA methylation. This causes enzymes inside a cell to attach a minuscule molecular compound, a methyl group, to a gene. This compound can deactivate or at times activate the gene, but the gene remains. The cell's DNA profile is unchanged. This could explain the difference between identical twins Jake and Tom. Some as yet unidentified environmental factor in early cell development has caused a methyl group to attach itself to a gene on one of Tom's chromosomes, a gene associated with growth. The growth gene is still there, but it's been switched off. By investigating the mystery of twins like Tom and Jake, geneticists are finally beginning to find clues to some of the great mysteries of the human genome. This is a map of chromosomes belonging to identical twins from age 3 on the left to twins aged 50 on the right. The red markings are an indication of the increase in the methylation that has occurred to their chromosomes over half a century. We know that factors like smoking, diet, and chemical exposure can, over a lifetime, wreak havoc with how genes operate. What's astounding is that there are different environmental influences in not only our lifetime, but also in utero. Hormones, space, quality of nutrition can all affect identical embryos differently, even those sharing a placenta. The activation and deactivation of genes during early development could explain many of the twists of fate that affect us all. Why one person is struck by disease while another spared. Epigenetics may also play a significant role in determining sexuality. If sexual preference is associated with an as yet unidentified gene, it may be that the epigenetic suppression or activation of this gene dictates sexual preference. These genetic switches may be the answer to why one twin absorbs more testosterone than the other, resulting in one being gay and the other straight. It's becoming clear that our health, personality, tastes, and even appearance are not the product of either our genes or our environment, but that nature and nurture are inextricably bound, with epigenetics the tangible biological link between the two. And most surprisingly of all, new research claims these subtle changes to a gene's expression can be passed on down through the generations without affecting the underlying DNA. Studying a register of births and deaths in a remote region of northern Sweden, researchers noticed a peculiar phenomenon. A generation of boys who experienced a famine had grandchildren who led longer lives. On the other hand, descendants of those boys who enjoyed plentiful food as children experienced an increased risk of diabetes, cardiovascular disease, and higher mortality rates. The effect of famine was especially strong if the boy was at the age when he was just about to start producing reproductive cells, his sperm. It seems the genetic makeup of these reproductive cells were directly but subtly altered by the famine or feast. Through their sperm, they passed on a nutritional legacy to future generations. The more we explore the hidden world of epigenetics, the more it seems we are not simply a product of our genes. We may even have the power to steer our genetic destiny, and perhaps that of our descendants, through lifestyle choices such as smoking, diet, and exercise. What makes identicals unique and useful to genetics is that their genome is a 100% match. Or at least, that's what we thought. Now, powerful new DNA techniques are revealing a different story. one that is only just unfolding. As the second trimester draws to a close, the twins will have a second scan. The development of external features such as limbs and eyes will be checked, and the major organs will be examined for signs of defects. The twins are about 20 centimeters long, small enough to cradle in the palm of the hand. The 20-week scan is the time when most mothers will discover whether they're having a boy or a girl. Gender identification can be done as early as 11 to 12 weeks, but is more certain at the 4-month scan. The scan reveals our twins are boys. Our twins are 24 weeks into development. Their mother can now feel them kick. Inside the womb, a battle for space is raging. A scan reveals this struggle, and perhaps even a glimpse of emerging personalities. Ultrasound studies, combined with observations of twins after birth, show very early forms of shy or extrovert behavior. In one study of fraternal twins, the shyer twin would occasionally hang on to his umbilical cord and even appear to lick it, while his sister would seek to grab, kick, and push him. Intriguingly, this pattern continued after birth. At 4 years of age, the shy twin still exhibited retiring behavior, while his more extrovert sister would try to grab and play with him. It seems our personalities may well begin to form in the womb. From week 29 onwards, the fetuses are beginning to resemble newborn babies. Their body fat rapidly increases. Bones are fully developed, but still soft and pliable. Head hair grows thicker, and fingernails reach the fingertips. Already, our twins' fingerprints are different. Epigenetic changes have already impacted the genes that control the pattern of the fingertips. These fingerprint differences will become more pronounced. As the fingertips develop, they receive blood from the veins. This makes them swell, forming distinctive fingerprint patterns. If blood flow from the placenta in one twin is restricted, blood may be directed away from the fingers and other extremities, leaving this twin with more whorls on their fingerprints than the other. Our fingerprint patterns can help to indicate how unhealthy our heart will be in adult life. The more whorls, the greater the chance of later heart problems. From the moment a fertilized egg divides in two, various influences on the genetic code conspire to create differences between identical twins. Differences that can become more pronounced over time. Yet it's always been assumed that identical twins share identical DNA. Until a discovery in 2007 surprised the scientific world. Researchers set out to investigate one of the most puzzling anomalies in the life of twins. Why one succumbs to a genetically inherited disease while the other is unaffected. They chose Parkinson's because it's more likely to be caused by a spontaneous genetic cause than be inherited. So the scientists examined the genes of 19 pairs of identical twins, including 9 pairs in which only one twin showed signs of the genetic disorder. They wanted to challenge the common perception that identicals are genetically identical. Indeed, their analysis showed that there were differences between the number of genes each twin had. Genes usually occur in 2 copies, one inherited from each parent. But this study has revealed it's possible to have just 1 copy, or to have 3 or more copies of the same gene. This phenomenon, called copy number variation, can fundamentally alter the gene's function and may induce the disease in one twin and not the other. It's a major breakthrough in our understanding of disease and in the study of twins. Because the discovery means that identicals are not a 100% genetic match. The very definition of identical twins needs to be revised, as advances in technology can prove that small but significant changes are present in all identical twins. Our identical twin boys are approaching full term. Already there will be subtle genetic differences between them that may shape very different futures. But first, they must face the most dangerous stage of their long journey in the womb, the birth. It's 35 weeks. The twins are now fully developed. They have hair, eyelashes, nails, and can open and close their eyes and mouths. Conditions in the womb are becoming cramped. At around 2 kilos, each weighs about 1/3 less than the average singleton baby. But combined, they put tremendous pressure on the womb. Premature deliveries for twins before full term at 37 weeks are common. Our twins will be born at week 35 by caesarean section. About half of twins are delivered this way due to the difficulties and dangers of giving birth to 2 babies naturally. To ensure safety, there is a larger than normal delivery team on hand. The obstetrician swiftly cuts the first membrane and then the second. Caesareans are always performed quickly to reduce the risks of complications. He plunges his hand into the womb and takes the first baby out. The baby takes his first breath and emits a healthy scream. The cord to the placenta, his lifeline for the last 35 weeks, is finally cut. Working briskly, the surgeon looks for his twin. With his twin brother out of the way, he follows quickly, also head first. Following birth, it's not always obvious to the obstetrician whether the twins are identical or fraternal. Our twins have a shared placenta, the clearest indication that they are identical. If the identicals have separate placentas, as happens occasionally, DNA analysis is the only sure method of identification. After an extraordinary journey, our two reproductive marvels have made it safely into the world. They may look alike, but we now know they are already different. "245 days ago, a fertilized egg divided, creating two identical embryos. From that moment onwards, their paths began to diverge. Inequalities in nutrition or space, differing exposure to hormones, small alterations in their epigenetic profile, even minute differences in the underlying genome, all combine to create subtle yet significant differences. Now, the twins are about to embark on the next stage of their journey. As they grow, facing diverse experiences and influences, the differences between them will become ever greater. These differences bring us closer to answering some of humanity's most profound questions. The study of identical twins has shed new light on the complex mix of genetics and environment, nature and nurture, that makes each of us unique. It has profound and far-reaching implications for how we all lead our lives.